Another method of activate and deactivate is referred to as the feedback inhibition. In this case, we need to understand that enzymes do not function alone independently by themselves. And here we have to define for you a reaction pathway, which is made up of lots of enzymes that work together. The product that's made by the last enzyme is called the final product, and this final product may actually inhibit an enzyme upstream or closer to the beginning of the entire process. To illustrate this, we'll use these symbols to show multiple enzymes. So this enzyme 1, enzyme 2, enzyme 3, all the way up to enzyme 20, didn't draw all of the other ones in between, represents what we call a metabolic pathway. Metabolic pathways are made up of groups of enzymes that all function together to turn some initial substrate into a very specific final product. It's more realistic in our organelles and in our cells that metabolic reactions occur where enzymes are grouped together into functioning units called metabolic pathways. Each enzyme will take the substrate, convert it into a product, and the product will become the new substrate for the next enzyme. So all of these enzymes can be floating together in the same organelle. Some of them may be bound to the surfaces of some of the membranes, but no matter how they are arranged inside the area where the reaction occurs, they're all available to the substrates that are produced by the previous enzymes. So here, initial substrate is turned into substrate 2, and we consider every substrate in between to be an intermediate product of this pathway. The only thing we're concerned about in the pathway here is the final product. In reality, intermediate products can become the primary or initial substrate for other metabolic pathways, but we'll discuss that another time. So now what will happen to substrate 2? Substrate 2 is now the right shape for active site of enzyme 2, which will then produce substrate 3. In each of these reactions, the enzyme is only capable of doing very little to each substrate. So, for instance, the initial substrate, when it enters and leaves, maybe only one bond is made or broken, or maybe only one electron is exchanged. So I want you to remember that about each enzyme. Each enzyme is only capable of very little modification to each substrate. So here, since we've been through 2 steps, substrate 3 may have only had 2 minor modifications made compared to when it entered as substrate 1, or initial substrate. It continues as enzyme 2 produces product, or intermediate product 3, or substrate 3. That will enter enzyme 3, producing substrate 4, and so on and so forth. Once you get all the way through to the 20th enzyme, the last intermediate product enters the last enzyme, and out pops the final product. Final products are usually immediately used by a cell. Once that cell has used the final product, it's going to then be in absence. So now we're waiting for the next final product to be produced by the end of this pathway, and then again it'll be used by the cell. This will continue as long as initial products are given to the beginning of the pathway upstream, and final products are produced at the end of the process downstream. But what happens if the final product is made faster than the cell is able to use them at that particular moment? Then here, what will occur is an excess of final product will start to show up inside the cell. Sometimes this occurs, and this is wasteful. If we continue to use valuable resources and convert them into products when we don't necessarily need them, we could end up exhausting supplies of important resources. So we can't have this continue and have lots and lots of excess final product made, and this could end up eventually killing the cell. So feedback inhibition is a method where these final products can actually act as an inhibitor for an enzyme upstream. In other words, when there's too much of the final product, the final product can become an off switch for the process to make the final product. So here we see it showing the excess final product working as a competitive inhibitor of enzyme 1. Once attached to the active site, no more initial substrate can enter the active site of enzyme 1, therefore no more intermediate products are made, and also no more final products are produced. So now the cell is going to continue to use final product, and whatever is made in excess will slowly be consumed. As the cell uses more and more final product, we'll see that the concentration will begin to diminish. And once the last molecule of excess final product is used, there's still one more. The ones that are now acting as inhibitors will diffuse from where they're more concentrated, which is in the active site of the enzymes, into the liquid of the cell, which is where it's less concentrated, and eventually it makes its way to where it's used up in the metabolic reactions. So what will happen once this inhibitor is removed? Now initial substrates are able to enter enzyme number 1 again. That will result in the metabolic pathway reestablishing. Final products will be produced once again, and then now the cell will have a new supply of final products to be produced. This will continue, of course, until more final product is made than can be consumed, and then the negative feedback system will work again to modify and control how much of the final product is made. The point of this is that lots and lots of enzymes then can be controlled not independently, but in a metabolic pathway it can control 20 enzymes by controlling only one. And in this case, what the metabolic pathway produces actually acts as the control method for the process itself. So this can help explain away a great deal of complexity in metabolism, and this is a completely automated process happening by random collisions without any kind of thought process or conscious ability necessary to make it work. The last method of controlling metabolism is referred to as localization and containment. Separate enzymes for a specific reaction pathway are separated from other enzymes and/or resources. This is the importance of compartmentalization, or having internal cell membranes within a cell. By controlling where the enzymes are and where the reactants or substrates are, we can separate and prevent reactions from occurring until we want it to occur. So by containing the reactants away from the enzymes, we can turn the reaction on and off by allowing the two to come in contact with each other. Some enzymes are attached to membranes so that they can be on the outside or the inside of a membrane, and then the reactants or substrates can be held on the other side until you want the reaction to occur. Some enzymes are not attached to membranes. Instead, they're floating free in the cytosol within a membrane space or within a membrane vesicle or vacuole. And in those organelles or bubbles, these enzymes can be isolated from their reactants or substrates. And if they're brought together into the same container, then the reactions can occur. So when we look at organelles, we should think of them as ways to actually isolate and separate one metabolic reaction pathway from another. Tissues and organs are made of different cells, which contains different concentrations or collections of organelles. And because different metabolic pathways carry out different functions, this is how one organ or tissue can have a completely different physiological function than another. Thus, when you stack metabolic pathways below the level of organelles, which comes under the level of cellular organization, which comes under tissues, and then organs and organ systems, we then define a hierarchy of organization for all biochemical or metabolic reactions.